Right, okay. Right, let's look at what is written on the board or what's in your notes for the stages of oogenesis, and then maybe we can make some amendments to the diagram or give clarification on the diagram. Okay, so basics, oogenesis, process for which eggs are made. First thing, so distinctly different to the spermatogenesis, is that the process starts in the fetus. It doesn't wait till puberty, it starts in the fetus. So within the fetus, the germ cells lining the ovary, there's the ovary, germ cells, we're talking about these, they divide by mitosis to form Oogonia. Right. I cannot think, I've never seen it, and I cannot imagine them ever asking you just to distinguish between, I've said this for spermatogenesis as well, germ cells and oogonia. So I can spend a bit of time now saying that the difference between the two. So when we start with germ cells, and then they'll undergo mitosis, a degree of specialization then, and they form oogonia. And then and at the same time, what you get is one cell divided by mitosis and producing two daughter cells, one of which will specialize somewhat to become oogonia, and the other one will replenish what was there to begin with, so to keep that layer of germ cells there. But to be honest, I'm pretty confident to say, do you know what? Germ cells, oogonia, same thing. To all intents and purposes, treat them as the same thing. If anyone was here was thinking, I'm a little bit unsure about this difference between oogonia and germ cells, sod it, oogonia then. Just call them that, they're the same thing. And then things, it's a level of complexity that we don't need to deal with. The reason I kept it in there, so I'm tempted to say, do you know what, sod that. The process begins in the ovary when the outer layer of the ovary, oogonia cells, divide by mitosis to form two subpopulations, the primary oocytes and follicle cells. So that's it starting there. I think you'll almost be fine. Almost certainly, sorry, you'll be fine. But okay, that's what we mean by the outer layer of the cells of the ovary. So now let's go to the oogonia. So we have many oogonia. And the next stage then is the oogonia divide by mitosis to form two subpopulations. Now that sounds odd because you're saying we've got one cell and we're going to form two different cells from it. Now what's going to happen is we're going to divide by mitosis to form two cells, and that will continue to happen and continue to happen. We'll have a number of cells, and of those cells, the vast majority will specialize somewhat in forming follicle cells. And one of them will specialize in a different way and form a primary oocyte. And the primary oocyte will be bigger, that will be part of its specialization. And the rest, the follicle cells, relatively small. So these are the follicle cells. Now the problem with that, the sticking point, you say, well hang on a minute, mitosis, I'll write it on there, mitosis should result in surely in forming identical cells. Yes, but then there's a degree of specialization which we end up with two subpopulations. We have primary oocytes and follicle cells. Those combine, or rather they associate. So I'll stick to the colours in the diagram there. So we've got primary oocyte, okay, in blue, and then the follicle cells. So they associate in that we have that primary oocyte then being surrounded by those follicle cells. And that collective structure now is a primary follicle. So a primary follicle is equal to a primary oocyte surrounded by follicle cells. That is the definition of a primary follicle. Okay, so it's all come from oogonia. Two subpopulations associate, form a primary follicle, and that is the primary oocyte surrounded by follicle cells. So, it says at birth, a baby girl will already have a large number of primary follicles. So, at birth, you're looking at, say, between 500 and 1,000 of these in your ovaries, in a girl's, in a girl's ovaries at birth. And that's it. That's all the would-be eggs, because they're not eggs yet. That's all the eggs, I'll use now in inverted commas, that you're ever going to have. Now, they then start to undergo meiosis, all of them. But they stop at prophase 1 of meiosis. Okay, and they stay there. So at birth, I say they then undergo, so at birth you are born with 500 to 1,000 primary follicles which have started, okay, I'll say which have started to undergo meiosis, I should be clear, within which the primary oocyte has started to undergo meiosis 1. So it's not quite right to say the primary follicle is undergoing meiosis 1, because the primary follicle consists of 2 subpopulations. Really, it's the primary oocyte which is beginning to undergo meiosis, and it stops at prophase 1. So at birth, you've got 1,000 of those-ish halted at prophase 1 of meiosis. And then things take a back seat for a while until you hit puberty. So, onset of puberty, production of hormones from the pituitary gland, that the hormones we'll come on to later, we'll talk more about later. What happens is each month from puberty, we often will say that a number of primary follicles may begin to develop, but only one will go on to fully develop. So let's just deal with that one. So onset of puberty, once a month, one of these primary follicles is going to develop further. Now this is the difficult part really, because there's 2 aspects going on here. There's the development of the oocyte itself, what I've got in blue, and there's the development of the follicle as a whole, which is all of this. There's 2 separate things going on here, and they happen alongside each other. But we kind of need to deal with both in isolation first. So it says, so each month, one primary oocyte completes the first meiotic division to form a secondary oocyte. So what do we mean by that? Let's now just show what we're expecting to see here. So we start with the primary oocyte, which is that. Okay, and now that is inside the primary follicle. It undergoes, let's complete it. When it undergoes meiosis 1, it will form a secondary oocyte, which I'm going to denote as being different in red, and it forms a polar body. So what do we mean by this polar body? Well look, the norm is we start with, I'll just go a little bit further. Then they undergo meiosis 2, and to form a mature egg or ovum and another polar body. Now all of that takes place within the follicle. Okay, it's not perfect that. It's not perfect for a reason we'll come on to later. Because once we get ovulation, we can argue it's out of the follicle. Hello? Hi. Can I call you again? Is that right? I'll call you at 10ish. That's okay. Lovely, thank you. Bye. So all of this here, that's your normal meiosis, really is going on inside the follicle. Okay, now, there's a little bit more to it than that because part of this happens after ovulation and there could be some argument whether it's still within the follicle. But okay, we'll come back to that later on. Let's just sort the this issue out here of what's going on with this polar body. Okay, so there's our second polar body there. So frequently get asked, what's the point of the polar body? Well, the point of the polar body is it gets rid of the other DNA that we don't really need. So really what we want here is a haploid cell with half of the DNA content as our original cell. Well, normally what happens is we have meiosis 1 and meiosis 2, and we end up with 4 daughter cells. But we don't want 4 daughter cells, because we don't want 4 eggs being produced at once, because we don't want 4 offspring being produced at once. So these are not necessary. So the way of the body in humans, because I imagine it's different in organisms where they can handle multiple births much better, is that we still have to undergo the process of meiosis, but the DNA that's been separated here has to go somewhere, and it goes into the polar body. Now, it's an uneven split in the sense that the DNA is split evenly, but the cytoplasm is not. So all the cytoplasm stays with this now secondary oocyte, because that's got all of the things like the mitochondria, the growth factors that are needed to stimulate growth once fertilization takes place. Same here, all the cytoplasm goes into the egg, and the polar body's just got DNA. So it allows us sort of to get rid of these. So we don't produce 4 eggs, we only produce 1, which is appropriate in humans. So that's the reason why polar bodies are produced. Right, so I said we're here right now. So at puberty, hormones produced by the pituitary gland stimulate the follicle to develop further, okay, full stop. So each month, 1 primary oocyte within the follicle completes its first meiotic division to form a secondary oocyte and a small polar body. Just hold fire for a moment. Just remember that what happened is we suspended, so we had a primary oocyte, it got suspended at prophase 1 until puberty. So I don't know, until puberty it should be there. And then the rest carries on. So puberty, this happens, oh, now we can carry on, and we get to this stage. And it then says at the end of that point, it is now referred to as a Graphene follicle. It's a bit tricky, I guess, not perfect. Let me just establish what we mean by. So we've now moved away, we just talked about the oocyte. The primary oocyte undergoes or completes meiosis 1 and forms a secondary oocyte in the polar body. Forget the polar body now. That will break down, that will degenerate, not relevant anymore. We have a secondary oocyte. And then we say it's now referred to as a Graphene follicle. Now that's not perfect. Let me show you what a Graphene follicle looks like and show you how it develops from a primary follicle. So this doesn't necessarily, so the difference between a Graphene follicle and a primary follicle, and indeed a secondary follicle we could refer to as well, is just the stage of the development. It doesn't necessarily, the stage of the development of the follicle cells, or the follicle as a whole, not necessarily to the state of play with the oocyte within it. But let me show you what I mean. So what happens to these follicle cells is they all divide by mitosis 2. So whilst the oocyte is developing and undergoing meiosis 1, and indeed meiosis 2, meiosis 1, is these follicle cells will be divided by mitosis. So that's what we started with. I'm doing it smaller for a reason. But we start to get more follicle cells. And we start to form a fluid-filled space. Okay, so I hesitate now to label that as a primary oocyte, using the same colours there. Should I be calling that a secondary oocyte? That's a little bit more tricky because they are independent. The development of the follicle happens independently, or what we call that follicle happens independently to what's happening in the oocyte itself. So I hesitate, in fact, for that reason I'm going to hedge my bets and say, look, it doesn't matter, it might be the primary oocyte, it might be a secondary oocyte. What we can quite safely say is that when this develops further and we get to a mature Graphene follicle, so I'm going to do my very best to draw a mature Graphene follicle here, which may take a little bit of time. So what I've done is I've just enlarged that fluid-filled space. And I'll just add to that a bit more to make my next point clearer. That is without doubt a Graphene follicle. Now what we will know about the Graphene follicle is it will have a secondary oocyte within it. It will have a large fluid-filled space. It's very distinctive. You may well be asked to identify a Graphene follicle, and it's very distinctive. They would not ask you to identify, I don't think, a primary follicle even. Okay, so we could argue that this is a secondary follicle, and I have seen it referred to, not in an exam, but I've seen secondary follicles referred to. I would say virtually zero chance of you being asked to do that. A very small chance to identify a primary follicle, but it's so small that it's quite difficult for them to show that image, a microscopy image, and say, what's this? There's a very good chance you're going to be asked to identify that. Okay, and you'll see when you see a picture of a Graphene follicle, well, if they're asking you to label it, it's almost certainly a Graphene follicle. So what's going on here? What's happening here? Well, look, the Graphene follicle migrates to the surface of the ovary. So the ovary, I draw the ovary on, and the ovaries are oval. That's the ovary, okay? In fact, the ovary would be significantly bigger than that, but it doesn't matter. The primary oocyte, the secondary oocyte now, still got some follicle cells surrounding it, is on the surface. It's on the outside of that ovary. But look here. This needs to be released. Ovulation needs to happen. So how are we going to make that happen? Well, what we've got is really just one adhesion point on either side. So what we've got to do is we break that and break that. This is loose. Now the reason I added the more cells on here, you see we've got multiple adhesion points here and here. So releasing that is tricky. This is all about being ready for ovulation, this Graphene follicle. We've got this fluid-filled space there, which I'm sure has got more, the fluid has probably got more of a role than this. But what it's doing, you can imagine, even if you can think of it like a pressure system, that the fluid that's being secreted in here by the follicle cells is building up pressure and is starting to, or is moving towards this bursting, and the secondary oocyte, surrounded by some follicle cells, they are, being released. And that's ovulation. The point at which that is released is ovulation. Now, we're kind of moving to two different things there. This is the level of the oocyte. This is the level of the follicle. How do they marry up? Well, what we know is a primary follicle has definitely got a primary oocyte in it. A Graphene follicle has definitely got a secondary oocyte in it. But you can't, I don't think in your notes it makes any reference to a secondary follicle, but you can't actually say, oh, secondary follicle, secondary oocyte. No. In fact, I think I'll just take it off. Look, the follicle develops, and at the same time, the oocyte is undergoing its, well, meiosis 1. But they're independent of each other. So you can't have some rule of saying secondary follicle means secondary oocyte. No. Well, the rule is Graphene follicle, secondary oocyte. Primary follicle, primary oocyte. That's it. And you're not going to be able to distinguish any further. Right. So this has been released now. This, let's do it over here. So now we've got, now being released at ovulation, we've got a secondary oocyte surrounded by some follicle cells. There are a couple of other layers there we could refer to, but now's not the time. So that's our secondary oocyte there. I should add, I could add on here, certainly here, if it's a secondary oocyte, by this point the polar body has been released. So if I put that in green there, polar body, where would that polar body be? The polar body's there. It's still within that structure here, and therefore it would still be in here. But in time it will degenerate, and it's therefore, I think I prefer just to leave it out, but because it just plays no role. But there it looks like they separated, and they have, but they're still part of this bigger structure. So secondary oocyte is released at ovulation, surrounded by some follicle cells. So now where we are is here. So okay, well now we need meiosis 2 to take place. Okay. So first thing that's I think of note is that what we tend to say in sort of common usage is that each month an egg is released from the ovary. Well, that's not right, is it? That each month a secondary oocyte is released from the ovary, because that's the egg, and we're not there, we're there. So each month a secondary oocyte is released from the ovary. In order for meiosis 2 to take place, we really have to get, and I say loosely now, we need fertilization. Okay, now that's wrong to put it like that, but certainly we need the sperm involved. And I'll show you exactly what needs to happen. So it's a little bit weird, because what it's saying is that if you're a woman who you're in your 90s, you're on your deathbed, and you've never had sex, you will never have actually made an egg. All you would have made is secondary oocytes. And you've released those every month, but because you never had any sperm deposited in your reproductive tract, this never happened. So you never actually produce any eggs. And that's an odd idea, because we think, oh, surely the idea is you release an egg each month. Well, we're completely wrong with that. No, it's secondary oocyte. Now, I could say, well, if you never had children, but no, because actually it's possible that you may have had sex, this stage may well have happened, but it never developed or you had a miscarriage, and therefore you never had children. Okay, so I've got to say that's why I said if you've never had sex, then that's the case. So this is what's released. How do we go about getting meiosis 2? Well, in order for meiosis 2 to actually take place, I said just now we need fertilization. I said that's not quite right. Right, fertilization is defined as fusing of the nuclei of the gametes. Okay, now that is not needed. In fact, that cannot happen before meiosis 2 takes place. Meiosis 2 has to happen first. So specifically what has to happen, let's take that out a minute. So what we have to get is there's a sperm, yeah, and inside, let's take a different colour. So I've got the nucleus of the sperm there, and I could try and draw a nucleus here, but it doesn't quite work, because what we've got is DNA that hasn't undergone meiosis 2 yet, but I need to get that nucleus in there. So what happens is the sperm comes into contact with the secondary oocyte, and then the membranes fuse. So at that point there, that fusion of the membranes, so we can say this a bit more crudely, say when the sperm penetrates the secondary oocyte, okay? But better, when the membrane of the sperm and the membrane of the secondary oocyte fuse, at that point, meiosis 2 is stimulated. Then that happens, which is why I say, if you've never had sperm in your reproductive tract, that cannot happen. Meiosis 2 cannot take place. So what happens then? Then meiosis 2 takes place. It will result in the production of a second polar body, okay, this one here, there it is, second polar body, and now that final nucleus, or that haploid half DNA of the egg. Now the nuclei can fuse, and I'll draw it together now in one go, and then, so when these nuclei fuse, I'll do it slightly bigger, nucleus of the sperm fuses the nucleus of the egg, that now is a zygote. That is fertilization. That is fertilization because the nuclei are fused. So sperm meeting the egg, that's not fertilization. Nuclei fusing, that's fertilization. Doing so, we've now formed a zygote. Okay? Last thing on this point, whatever's left, this thing here, the remainder of the Graphene follicle, that develops into a structure called the Corpus Luteum. Latin aficionados, corpus, body, luteum, oh, yellow. Okay, I say okay, like, oh, remember that, who cares? You need to know the name, okay, Corpus Luteum. Now the Corpus Luteum has got a role later on. Really, the role, actually it's an interesting point now. The Corpus Luteum has got a role in the immediate secretion of the hormone progesterone. Now we will look at hormones later, and we'll see this in more detail, but the important point here is progesterone is responsible for maintaining the lining of the womb. So without progesterone, if progesterone levels drop, what you get is menstruation. So really that's saying progesterone levels have dropped, saying there's no pregnancy, shed the menstrual lining and restart the menstrual cycle. Okay, now what happens, and I'll just put it very briefly, and we'll do it later, the zygote, or well, then forms an embryo, and the embryo secretes a hormone called HCG, human chorionic gonadotrophin. We'll look at it again, but HCG. And HCG causes that to persist, the corpus luteum to stay as a corpus luteum, not degenerate, and causes it to continue to secrete progesterone, and therefore causes the lining of the womb to be maintained. Why? Because we've got pregnancy. Zygote produced makes this, which ultimately means that stays being produced, which means the lining of the womb maintains because that needs it. In fact, that is what the pregnancy test detects. That's what they're looking for, your antibodies. I think it's still antibodies in a pregnancy test. It's looking for HCG. If it sees that, it's a positive, obviously then, positive pregnancy test.